the past few decades, plastic materials have been used increasingly in food, clothing, shelter, transportation, construction, medical, and recreational industries. Plastics are advantageous as they are strong, lightweighted, and durable. However, they are resistant to biodegradation, leading to the pollution that harms the natural environment. The successful production and marketing of biodegradable plastics will help alleviate the problem of environmental pollution. Several companies started introducing biodegradable plastics into the market. However, none of them is efficiently biodegradable in landfills. For this reason, none of the products has gained widespread use. Hence, there is an urgent need to develop efficient microorganisms and their products to solve this global issue. Polythene is one of the most commonly found non degradable solid wastes, recognized as a threat to marine life. It can cause blockage in the intestines of fish, birds, and marine mammals. The degradation of polythene poses a challenge since it is used often. A very general estimate of worldwide plastic waste generation annually is about 57 million tons. This solid waste related problem poses a threat to megacities, especially coastal ones. Coastal mangroves were historically favored dumping sites for solid waste disposal. This paper attempts to isolate potent microorganisms that degrade the plastic materials from the mangrove sediments.
Plastic cups and polythene bags were buried at a depth of 5 centimeters in the mangrove soil under two zones, colonized by Rhizophora sp and Avicennia sp, along the Villiers Estuario of 11° 29 north, 79° 46 east, southeast coast of India. The materials were allowed to degrade naturally in the mangrove soil, and they were sampled at intervals of 2, 4, 6, and 9 months using sterile forceps and transferred to laboratory aseptically. One set of samples was thoroughly washed using distilled water, shade dried, and then weighed for final weight. The degradation was determined in terms of percent of weight loss of the materials over a period. Another set of sampled materials was washed gently using sterile water to remove soil debris. About 1 gram of the materials infested with bacteria and fungi was transferred into the conical flask having 99 ml of sterile water. This content, which was shaken vigorously for its equal distribution, was serially diluted. The pour plate method was adopted using the Zavos agar medium for bacteria and the Martin Rose Bingle medium for fungi. For each dilution, 3 replicates were made. The plates were then incubated at 30 degrees Celsius for 2 to 7 days. The bacterial and fungal counts were then made. The dominant ones were isolated and subcultured repeatedly for getting pure colonies and then preserved in slant tubes for further identification. The bacterial strains were identified based on the case dated by Oliver of 1982. The tests conducted were motility tests, glucose oxidation, penicillin sensitivity, and glucose fermentation for gram-negative bacteria, and dextrose fermentation, catalase, and glucose utilization for gram-positive bacteria. The fungal strains were identified after straining them with cotton glue by following the keys of Rubber and Fennell 1987. To assess this, the pre-weighed disc of 1 cm diameter prepared from polythene bags and disposable plastic cups were aseptically transferred to the conical flask containing 50 ml of kosher broth medium, inoculated with different bacterial and fungal species separately. Nutrient broth medium was used for bacteria and rose bingle broth medium for fungi. Control was maintained with plastic discs and the micro-free medium. 4 flasks were maintained for each treatment and left in a shaker. After 1 month of shaking, the plastic discs were collected. Wash thoroughly using distilled water, shade dried, and then wait for final weight. From the data collected, weight loss of the plastics and polythene bags was calculated.
Under the soil conditions of two mangrove species, an experiment was designed to degrade polythene bags and plastic cups. Table 1 summarizes the findings. The polythene bags deteriorated after 6 and 9 months of incubation in the soil, regardless of the mangrove zone, but not after 2 and 4 months. After 9 months of analysis, the plastic cups were shown to have designated, but not after 2, 4, or 6 months. After 9 months of investigation, the maximum biodegradation of polythene was 3.77% and 4.21%, respectively, under Rhizophora and Avicennia zones, while the comparable values for the biodegradation of plastics were only 0.25% and 0.17%. The microbial counts on the polythene bags and plastic cups that degraded for different months under two mangrove zones were recorded. However, there was no statistically significant variations of bacterial counts between the two mangrove zones, which is the Rhizophora and Avicennia, on or between the months of degradation.
The microbial species identified from degrading polythene bags were Bacillus species, Staphylococcus species, Streptococcus species, Diplococcus species, and Micrococcus species belong to gram-positive bacteria, Moraxella species, and Pseudomonas species that belong to gram-negative bacteria, and Aspergillus niger, A. ornatus, A. cremeus, A. flavus, A. candidus, A. ocraceus, A. nidulans, and A. glaucus belonging to fungi. Thus, even bacterial species and 8 fungal species were obtained. These microbial species were also recorded from degrading plastic bags except Bacillus species, Staphylococcus species, Aspergillus ornatus, A. cremeus, A. flavus, A. candidus, A. ocraceus, A. nidulans. There were 5 bacterial and 2 fungal species, commonly and predominantly found detected in both polythene and plastics, and these were selected for further study. 7 microbial species were tested in the laboratory for their ability of degrading the polythene and plastics. The species tested were Moraxella species, Pseudomonas, Staphylococcus, Micrococcus, and Streptococcus species, and 2 fungal species, Aspergillus niger and A. glaucus. These microbes were separately allowed to degrade the polythene and plastics under shaker cultures for a month. The results are shown in Table 2. Table 2 shows us the degradation of the polythene and plastics incubated with different microbial species in shaker cultures under laboratory conditions. Here we can see the name of the microbes and microbial degradation, percentage weight loss per month of polythene and plastics. The bacteria caused the biodegradation ranging from 2.9 to 20.54% for polythene and from 0.56 to 8.16% for plastics. Among the bacteria, Pseudomonas and Moraxella species were found most active in degrading 20.54% of polythene and 8.16% of plastics in 1 month period. Among the species, Aspergillus glaucus was more active than A. niger in degrading 28.8% of polythene and 7.26% of plastics within a month.
The researchers found that polythene is faster at biodegrading than plastics. Mangrove soil can be a source of factors for the degradation of plastic materials. Microbes is a factor besides moisture and etc. Microbial counts are high, favoring the degradation of plastics. Therefore, it has been experimentally proven that these microbes causes degradation of plastic materials up to 28.8% per month. The surface of plastic materials has turned from smooth to rough. Hence, further study on microbial enzymes or organic acids in degradation of the polythene and plastics will pave the way for finding technology for degrading the plastic materials which are otherwise hazardous to the environment. To summarize what we discussed earlier, the biodegradation of the materials was analyzed. Polythene bags scored higher than plastics. Microbial species found were 5 gram positive, 2 gram negative, and 8 aspergillus species. The efficacy was analyzed in shaker cultures. Pseudomonas scored 20.54% for the biodegradation of polythene and 8.16% for the plastics in a 1-month period. Aspergillus glaucus scored 28.80% of biodegrading polythene and 7.26% in plastics in a 1-month period. Thus, mangrove soil is a good source of microbes capable of degrading plastics. Thank you for listening, our dear classmates and our dear panelists. I hope you don't hesitate to ask. Thank you very much.